Hi everyone. Linkage, recombination, independent assortment. I think these are the terms that confuse most of the students in standard 12 biology. So what I have done is I have created one very simple, easiest explanation ever on this topic. So please watch it from starting till end, write notes and do comment if you have any doubts, and also comment if you want some numericals to be solved on the same. Thank you. So now first let us understand independent assortment. Let us imagine a dihybrid, capital A, capital B, small b. This is said to be dihybrid. Dihybrid, observe the name. Di means two. Di means two and hybrid means heterozygous. So here you have two heterozygous conditions. So we will call it as a dihybrid. So this is the chromosome arrangement in a dihybrid. As of now, we are talking about independent assortment. We are talking about independent assortment. Because once you understand independent assortment, only then you can understand what is linkage and recombination. So this is the case of independent assortment. Now two genes. Let us imagine two genes, capital A, small a, and capital B, small b. These are located on two different non-homologous chromosomes. This is said to be a homologous chromosome pair and this is another homologous chromosome pair. Now imagine this condition is represented like this. This is how we write and this is how it is there in the body in real, right? So in this case, whenever gametes are produced, so you have learned that a dihybrid produces four types of gametes, right? You have learned this formula probably, 2 raised to n, where n is equal to number of hybrid. So how many hybrids do you have over here? 1 and 2. So formula will be 2 raised to 2. So you will have 4 types of gametes over here. Now which 4? So whenever gametes are produced, you know that total 4 chromosomes are there. If 4 chromosomes are there, gametes will have half the number of chromosomes. So gametes will have 2 chromosomes. So those two will be, a pair never goes together in a chromosome. These two together never go in a chromosome and in the gametes, and these two never go together in a gamete. So what should be the combination? Now you will have this kind of combination. These two will segregate, law of segregation. These two will segregate, right? And now we will have one gamete with this combination. So that is why you are writing this capital A, capital B, and we may have one combination like this. So this is one gamete and this is another gamete. So you will have one combination with small a, small b. But do you think it is necessary that this chromosome should always go over here? Can't it happen that this chromosome went over here and this chromosome went over here? You will say, okay, yes, that is also a probability. So capital A and small b, capital A and small b, and small a and capital B. So small a and capital B. So what is the point over here is that here 4 types of gametes are produced. But more importantly, the main point is that they are produced in the ratio 1:1:1:1:1. That is the most important thing. That means that in case if there are 100 gametes produced, we will have probability of this 25%, 25%, 25%, and again 25%. That is the main point over here. And as capital B can go along with capital A also, and capital B can go along with small a also, the probability are almost equal, and that is the reason we call it as independent assortment. So once you have understood what is independent assortment, now we can talk about what is going to happen in the linkage and recombination, right?